the first structure that we are talking about is plasma membrane. Plasma membrane is the outermost membrane which is present in the cells. Plus, as we saw in the structure of eukaryotic cells, Golgi body is also made up of membrane, mitochondria also has membrane. So that membrane and this outer membrane, they are made up of the same things, that is proteins and phospholipids. The basic difference between the outer membrane and the membrane of the inner structures is the various ratios of proteins and phospholipids. So it could be more or less protein in some membrane, it could be more or less of phospholipid in some membrane. Plasma membrane shows certain characteristics. So we need to understand what all characteristics are shown by plasma membrane. So these are characteristics of plasma membrane. First, it is selectively permeable. We need to understand the meaning of selectively permeable. Say if we talk of a membrane which has pores, and this is say the outer side, this is the inner side, and these are some particles, various types of particles which are present here. If the membrane allows the entry of anything, everything which is depending upon the pore size. Pore size means this is the opening through which substances can go in. So anything which can pass through this pore, whether that substance is good or bad, whether that substance is useful or is a toxin, if it can pass this opening and go inside, then such a membrane will be termed as permeable membrane. So this is a permeable membrane. Second type of membrane is when, again there is a pore size, this gap which we have shown here is the opening or the pores which are present in the plasma membrane. Again let us see, there are various particles. So which particles would go in? Again which could pass through this. But plasma membrane, as we said, is selective. So what does this mean? Selective means the membrane is going to select which substances should go in and which substances should not. Say for example, we are talking about this particle. It is smaller, that means it can cross this pore. But if it is not required by the cell, it will not go in. This is a big particle. It cannot cross this opening, but if it is required by the cell, it will still go in, may not go through the pore, but may go through the other processes like endocytosis, but it will be taken. So the membrane selects what it wants or what the cell wants. So the plasma membrane that we are talking about is a selectively permeable membrane. This is one property which is shown by the plasma membrane. The second property is that the protein to lipid ratio is variable. That means it changes. In some places it could be 50-50, at other places it could be 60-40 or vice versa. And the third property which is shown by plasma membrane, that it is fluidic. One is dynamic or other is static. So here we are talking of the fluidy nature, fluidic nature of plasma membrane. So these three properties are exhibited by the plasma membrane. We are going to see the structure of plasma membrane as proposed by various scientists, and we will see which one satisfies all these three properties. The first model, there were about 100 models which were proposed by scientists. All of those models failed because of obvious reasons, that they could not explain the working of plasma membrane based on these properties. So first model which was accepted to a great level was sandwich model. Which was proposed by Danieli and Davison in the year 1935. This model is known as sandwich model. So as the name tells us, it's something sandwich-like structure. Before we draw the structure, let us see what all they told us about the membrane. They said it is made up of proteins and phospholipids. Secondly, they told us, the second point is that proteins which are present, they are globular proteins. That means spherical. So either the proteins are fibrous or they are globular. So here the proteins are globular. Third, the membrane is trilaminar. That means there are 3 layers. It is trilamellar. That means there are 3 layers. How are these 3 layers? The middle layer is of phospholipids and the upper and lower layers are of proteins. That means if we make just the middle layer, this is going to be the phospholipid, and there is one layer here, one layer on the outer side, one layer on the inner side. So now we see 3 layers, a middle layer of phospholipid, an outer and an inner layer. Now let us come to the structure. What exactly was the structure? They said that the phospholipid molecules, they make the middle layer. These phospholipid molecules have a head and 2 tails. It is actually glycerol attached to 2 fatty acids. So basically it is a diglyceride fat. So when we make this structure, we make the 2 tails and the head. The head part is hydrophilic and the tails are hydrophobic. Hydrophobic means hates water, hydro is water, and hydrophilic means have affection or affinity towards water. So this molecule, phospholipid molecule, has 2 ends, one hydrophilic head end and the other is hydrophobic tail end. Any molecule which has 2 opposite ends is known as amphipathic. So phospholipid molecule is amphipathic. This is very, very important. What is amphipathic? One molecule having 2 ends. One is hydrophilic and the other is hydrophobic. Now according to the structure, the phospholipid middle layer, this middle layer that we are talking about, is made up of phospholipid molecules, and these phospholipid molecules are arranged in 2 layers. The 2 layers are such that the hydrophobic tails face each other and the hydrophilic heads are on the outer side. So this complete is the phospholipid bilayer. On both the sides, outer side as well as on the inner side, there is a protein layer. As the proteins are globular, so we will draw circular proteins because globular means circular. So this protein layer is made up of globular proteins. This is on the outer side. Same would be on the inner side. So this is a protein layer and on the inner side also a protein layer. So trilamellar structure, as was told by them, that there are 3 layers, the middle layer of phospholipids and these 2. Does it look like a sandwich? If you have a bread and jam sandwich, the 2 slices of bread and we have a thick layer of cheese or maybe jam. So it looks like a sandwich, and that is why the name given to this model, which was the sandwich model. Plus it is also known as trilamellar model because of the 3 layers which are present. Few more points which were told to us by them was, one of them was that plasma membrane has pores. Pore size is 7 to 10 microns. This was also told to us by Danieli and Davison. So information given to us plus the model which was given to us. Let us see whether this model explains these 3 characteristics or not. If it explains, this model will be accepted, and if it does not, then it will get rejected. The first point itself says it is permeable, selectively permeable. That means small, large molecules can go through it. That molecule can be a water soluble. Suppose this molecule is a water soluble molecule. It should pass this plasma membrane. Because it is water soluble, it can go through this protein layer. Protein is hydrophilic, so there is no problem. When it comes to this head part, still there is no problem because the head is again hydrophilic. It will let the water or water soluble substances close. Now, what about this middle band? This middle band, which is made up of the tail region, hydrophobic. So now this particle which has reached here will not be able to cross this band of hydrophobic tails. So how can water soluble substance enter? If water soluble substance cannot enter, how will the cell survive? Because most of the things they enter as ions or in solution form. So this was the biggest drawback, that it could not explain the semi-permeability of the plasma membrane. The second point is protein lipid ratio is variable. You take this plasma membrane of any region. Suppose we talk about only this region, and let us count. This is only to understand the point. How many proteins are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 and 12. So there are 12 proteins. And how many phospholipid molecules are here? In this diagram we have 4. So the ratio is 12:4. How can this ratio change? In this model, it does not tell us anything about movement of these particles. So if these particles don't move, we cannot have other than this ratio. So it does not even explain the variable ratios of proteins and lipids. Coming to the third point. Fluidity means something can float or move on this membrane. This complete structure does not give us that idea of something floating. So this model, though it was one of the good models, you can say, because it was one of the first models which explained many things closer to the real structure of plasma membrane, but it was rejected on the basis of the structure not explaining the main criteria or characteristics of plasma membrane. Then came another model. This was the first one, that is the sandwich model. The second one is unit membrane model. Unit membrane model of plasma membrane. It was proposed by Robertson in 1952. This unit membrane model gave some variation from this. There are certain things which were common. Let us talk about the common points first. What was the common points, rather were common points? First one was that this is a protein and phospholipid membrane which was given by Danieli and Davison. It is a trilamellar membrane. same which was given by Danieli and Davson, 3 layers. Phospholipid molecules, are amphipathic. This is also common. The difference now is in the protein part. Here the proteins were globular. Robertson said that the proteins are fibrous. And one more additional information which was given by Robertson was the thickness of plasma membrane. The thickness of plasma membrane was given to be 70 microns approximately. Pore size again 7 to, where have we written the pore size? The pore size is same here, pore size is 7 to 10 microns. So every information was pretty much same except for this one. So now using this information, let us draw the structure which was given by Robertson. Again the phospholipids same, hydrophilic head on the outer side, hydrophobic tails on the inner side. This is the phospholipid bilayer. On either side the protein. Now because it is a fibrous protein, we will have to draw it in the form of a continuous fibrous structure. Looks like sandwich again, but it was called unit membrane. This middle layer is phospholipid, the outer is protein layer and inner is again protein layer. Let us see whether this structure explains the 3 characteristic features of the plasma membrane. First is selectively permeable, whether the membrane is permeable or not. It comes to the same point. Water soluble substance can reach up to here, but again it encounters a hydrophobic band. So this water soluble substance will not be able to travel through this hydrophobic. So entry of water soluble substances into the cell is impossible with this structure. Protein lipid ratio, if you again take a part of this, because these proteins and phospholipids are not moving from their place, the ratio is going to remain fixed. Third, fluidic nature, again it looks like a rigid structure because nothing is moving from its place. This is also a rigid structures. So both these models could not explain the basic 3 properties or characteristic features of the plasma membrane. So both were rejected. The third model was the accepted or the selected model. The third model, it has been accepted. We till date use the same model. It is called fluid mosaic model of plasma membrane. It was proposed by Singer and Nicholson. Let us first talk about the points which they put forth. First was, it is made up of proteins and phospholipids. So this is same which was given by all 3 models. Phospholipid molecules are amphipathic. This information is also same. It is a trilamellar structure. This is also same. So the differences are, the first important difference is that proteins, one, are globular and they are of 2 types, extrinsic and intrinsic. Extrinsic proteins are external proteins, intrinsic proteins are internal proteins. We'll come to the details of this. Phospholipid is same, so no change in this. Only the protein part is globular protein. Now let us draw the structure so that we understand the structure clearly. Phospholipids, same arrangement. Hydrophilic heads on the outer side and hydrophobic tails on the inner side. They said, Singer and Nicholson said that there are two types of proteins. Extrinsic proteins, globular, and they do not form a sheet. Intrinsic proteins can be of two categories. Some which are partially submerged and some which are completely submerged. That means they run through and through this phospholipid bilayer. So let us draw a couple of phospholipid molecules here. And the protein, globular proteins. Now, from this, let us take these criteria. First is selective permeability. Up till now, in the first two membranes, that is the sandwich model or the unit membrane model, the biggest problem was how would the water soluble substance cross this membrane? It can cross through this protein molecule. So these intrinsic proteins, they act as channels for water soluble substances. This is actually an intrinsic protein. So this big protein which we have drawn here is intrinsic. This partially submerged is also an intrinsic protein, and the ones which are on the outer side, these are the extrinsic proteins. And as we can see, these globular extrinsic proteins, they are not connected to each other. So there is no continuous sheet-like thing which is formed. Phospholipid molecules, same. So now water soluble substance can go through the membrane. So first characteristic proved by this model. Let us come to the second one. The second one says protein lipid ratio is variable. What Singer and Nicholson told us that the protein molecules, they float on the phospholipid bilayer, and that is why to this model they gave one more term. They called it protein icebergs on the sea of lipids. As icebergs float on water, similarly these proteins move from one place to another. Plus one more information which they gave was that these phospholipid molecules can also change their position. To understand this, let us number them. Say we are numbering the phospholipids of the upper chain as 1, 2, 3, 4, 5, 6 and of the lower chain as A, B, C, D, E, F and G. They show two types of movements. One movement is when the molecules interchange their position in the same layer. This is known as transition movement. So phospholipids show transition movement. What is meant by transition movement? The position of 1 and 3. So this becomes 1 and this becomes 3, gets exchanged. Movement is taking place in the same band. That means only these molecules can change their position, or only these molecules can interchange their position. The other type of movement shown by this is known as flip-flop. Flip-flop movement shows that phospholipid molecules interchange their position between the membranes or between the layers. So if this is 2 here and this is B here, B can come here and 2 can go here. So the position change is between the 2 layers. So there are 2 possibilities. These molecules can move between the same band or they can flip-flop between the 2 layers. Now if these kind of movements are taking place, then a situation might come. Say for example, again, let us take only this much band and see the ratio. Here there are 1, 2, 3, 4 phospholipids. So phospholipids are 4 and proteins are 2. So the ratio is 4 phospholipids and 2 proteins. But suppose this 2 moves from here and comes here. It takes this position. It goes here. The 2 comes here. What is here right now? There is nothing. So what has the ratio changed? There are 3 phospholipids and 2 proteins. One more possibility, that this protein, because it is acting like an iceberg, it is floating on this phospholipid, this protein changes its position and it comes here. So 3 phospholipids and only 1 protein molecule. So protein phospholipid ratio changes. And why is this protein able to move? Because the membrane is fluidic. It is like a jelly, so that something can move on it. Now this model can explain, or could explain rather, all 3 points satisfactorily. So this is the most accepted model. Till date we are using the same model of plasma membrane. Now as we are talking of plasma membrane, let us take the properties and few more things which are with the help of plasma membrane, like how exchange of substances takes place. We'll talk about osmosis, diffusion and few things related to that.